Today we're going to look at the central nervous system. So remember, your central nervous system is going to include structures that are encased in bone. So it will be the brain and the spinal cord. So if we look at the development of the central nervous system, we can see that there are three regions. The forebrain comes from the prosencephalon, the midbrain from the mesencephalon, and the hindbrain from the rhombencephalon. And so the prosencephalon will end up forming your cerebrum and your diencephalon. That's going to include the thalamus, subthalamus, epithalamus, and hypothalamus. The mesencephalon is your midbrain. Rhombencephalon is going to make your pons, the cerebellum, and the medulla oblongata. So your brainstem is a continuation of the spinal cord. This is going to come up from the spinal cord. It will turn into the brainstem and connect with everything else. So it will consist of the medulla oblongata, pons, and midbrain. The cerebellum is the second largest part of the brain. That's going to be this section in the back here. The diencephalon is going to give rise to the thalamus, hypothalamus, and epithalamus. It's pretty much right in the middle. And then the cerebrum is the largest part of the brain. That's going to be this over the top. So this is just another picture here or another chart showing divisions of the brain. Some of the things in the notes here are a little bit repetitive, but the idea is you pick which one is really clicking with you for your learning style to look at it. So this is a photograph of an actual human brain, a sagittal section, viewing it from the medial side. So physical protection of the brain is going to come from the bones of the cranium, as well as the cranial meninges, and then the cerebrospinal fluid. The idea is that you want to give it biochemical isolation. We do this through the blood-brain barrier. The brain is so critical to our function that you want it to be isolated from potential toxins that would get in the body or potential invaders. That extra layer of protection is helpful because the brain is so vital to the human survival. So for the cranial meninges, there's three layers. We call it a meninx if you're talking about it singularly. We've got the Dura Mater, Arachnoid Mater, and Pia Mater. And it's going to be continuous with the spinal meninges. This will help to protect the brain from cranial trauma. So it's kind of like a little fluid cushion around the brain. So when we look at the Dura Mater, this is the outermost layer. So the inner portion of it is going to have a fibrous layer that's the meningeal layer. The outer fibrous layer is the endosteal layer that's going to fuse to the periosteum that's on the inside of the bone. And then you'll have the venous sinuses between the two layers. The arachnoid mater is going to be in the subarachnoid space between the arachnoid and pia mater. It contacts the epithelial layer of the Dura Mater. And then the pia mater is the innermost layer. It's attached to the brain surface by the astrocytes. The dural folds are an inner layer of the Dura Mater. They're going to extend into the cranial cavity to create some divisions in there. It's going to help to stabilize and support the brain. They'll contain collecting veins like the dural sinuses. So what we have is the falx cerebri. That's going to separate the cerebral hemispheres. The tentorium cerebelli, that will separate the cerebrum from the cerebellum, and the falx cerebelli, that's going to separate the cerebellar hemispheres. So the brain receives about 20% of the total blood supply. An important set of vessels here are the internal carotid and vertebral arteries. These are going to end up forming the circle of Willis, so this blood flow will come up on both sides of the brain and meet on the other side. The internal jugular veins are going to return the blood from the brain. Our blood-brain barrier protects the brain from harmful substances. It's going to isolate the CNS neural tissue from the general circulation. This is going to be formed by using tight junctions that seal together the epithelial cells. So you'll have a continuous basement membrane. And then astrocyte processes are going to cover the capillaries. So these astrocytes will control the blood-brain barrier by releasing chemicals that will control the permeability of the endothelium. There are 4 areas where there are breaks in the blood-brain barrier. So portions of the hypothalamus that secrete hypothalamic hormones. The posterior lobe of the pituitary gland, it's going to secrete the hormones ADH and oxytocin. The pineal gland that will have your pineal secretions like melatonin. And then the choroid plexus, where you have special ependymal cells that will maintain the blood CSF barrier. So this here is an image showing the blood-brain barrier. Cerebrospinal fluid is going to surround all of the exposed surfaces of the CNS. This will interchange with the interstitial fluid of the brain. So there's 80 to 150 milliliters produced by the choroid plexus, or about 500 milliliters every day. It's a clear liquid. It's going to contain glucose, proteins, and ions. When we say clear, it's relatively transparent. If you do hold it up in front of a white piece of paper, you can actually see there is a slight color to the liquid. It's not clear like water. So functions, mechanical protection. The brain is going to float in there and it will help soften the impact against the bony walls. Chemical protection, it's got optimal ionic concentration for action potentials. And you should have a lot of those going on in the brain. And then circulation, nutrients and waste products can be carried to and from the bloodstream this way. So this shows the path of flow of the cerebrospinal fluid. So it can go from the lateral ventricles, choroid plexuses, to the lateral ventricles, through the inner vertebral foramina, down to the third ventricle, fourth ventricle, subarachnoid space, the arachnoid villi, and so on. You can see where there are other connections in here. So it is actually going to be somewhat continuous with the blood. It will have connections where it comes from the bloodstream and then will return to the bloodstream. So these ion concentration differences between the CF and the plasma are important. It helps support the CNS function like all of those action potentials. So it's going to be partially reabsorbed through the arachnoid villi. They're little grape-like clusters in the arachnoid villi that will penetrate the dural venous sinus. Some of it is also going to go back through the rest will actually go back through the venous sinus. So we have about a 20 milliliter per hour reabsorption rate, and it's the same as the production rate. That's important. You don't want to be putting more fluid into the brain than what is able to leave. Then you're just going to build up pressure, which will lead to other problems. So we have 4 ventricles in the brain. You can see the first 2 are lateral. The third ventricle is in the middle, and the fourth ventricle is also in the middle, just lower down. So the ventricles are CSF-filled cavities in the brain. The neural tube encloses the neural seal, and that will expand to form the chambers or ventricles that are going to be lined with ependymal cells. So lateral ventricles are in the cerebral hemispheres. Third ventricle is in the diencephalon. Cerebral aqueduct is in the midbrain, and then the fourth ventricle is down in the brainstem and cerebellum. So the medulla oblongata, this is going to allow the brain and spinal cord to communicate. It helps to coordinate complex autonomic reflexes and also will help in controlling visceral functions. So it's connected to the spinal cord, which will allow that communication. The pyramids are going to be bulges on the anterior aspect of the medulla. They're formed by the large corticospinal tracts that pass from the cerebrum to the spinal cord. So when you see the names of the tracts like this, they're usually named for where they start. So we're going to start in the cerebral cortex and go down through the spinal cord. So this is a common site for decussation. That's where nerves are going to cross over. And this will be decussation, a lot of the ascending and descending tracts. So in here, you can see this bulge, and you can see these different centers that you have in here. So these vital reflex centers are going to do your life, make the life-sustaining decisions. So they are important enough that the body doesn't give us conscious control over this. We're much better off with things happening automatically. So your cardiac center, this is going to control the rate and force of contractions. The vasomotor center, it's going to control smooth muscle in the blood vessels. So it will end up influencing blood pressure control and the diameter of the blood vessels. You have a respiratory rhythmicity center that will control the rate and depth of breathing. That will extend up into the pons. And then we have some non-vital reflex centers, vomiting, hiccups, coughing, swallowing, sneezing. So if a person has an upper cervical injury, sometimes they will have vomiting that will go with this. That is why. Cranial nerve 8 through 12 nuclei. The vestibular nuclei complex, this will help to subconsciously maintain your sense of balance or equilibrium. The solitary nucleus, this will receive visceral sensory information. The olivary nuclei, or sometimes called the olives, are lateral to the pyramids. They're going to receive sensory information from your proprioceptors in your skeletal muscle and joints and will act as a relay point to the cerebellum. So proprioceptors are going to be receptors that determine or send the message of where your body parts are in space relative to the rest of your body. So for example, if you put your hand behind your head, you don't have to turn your head around and look for it there. You can feel that it's there. That's proprioception. So a portion of the ventricle found here is the fourth ventricle. So pons means bridge. This is going to link the cerebellum with the mesencephalon, the diencephalon, the cerebrum, and spinal cord. So we have the nuclei of cranial nerves 5, 6, 7, and 8. We have nuclei involved with respiration. So you have an apneustic center and a pneumotaxic center that will modify respiratory rhythmicity center activity. The cerebellar peduncles connect the cerebellum to the brain stem. There are 6 of them in all. So the superior cerebellar peduncles connect to the midbrain. The middle cerebellar peduncles connect to the pons. They're the largest. And then the inferior cerebellar peduncles connect to the medulla. So the mesencephalon will extend from the pons to the diencephalon. So we've got the tectum, also called the corpora quadrigemina. Here you've got the superior colliculus that's involved with the visual reflex, the inferior colliculus with the auditory reflex. So the tegmentum, it's going to have the red nucleus. It's that way because it has a lot of blood vessels in it. And the substantia nigra, which is a gray pigmented area. And then the cerebral peduncles. So you have cerebral peduncles and cerebellar peduncles. The cerebral peduncles are going to contain descending fibers to the cerebellum. They're motor command fibers. So the substantia nigra is this dark pigmented area that's involved with subconscious muscle activity. This is the area involved with Parkinson's disease. The red nucleus helps with controlling voluntary movement of the limbs. And it will contain cranial nerves 3 and 4. So here's some more pictures of the mesencephalon. So a lot of times in the 200 level anatomy and physiology classes, you dissect a sheep's brain, and the sheep's brain is relatively small, making some of these structures a little hard to see. They're much easier to see on a full-size human brain. And so some additional pictures. So the reticular formation has scattered nuclei in the medulla, pons, and midbrain. It's part of the reticular activating system. This is going to alert the cerebral cortex to sensory signals, so the sound of an alarm, a flash of a light, smoke, an intruder. This is how your alarm clock wakes you up from sleep. It's going to help maintain consciousness and help keep you awake with stimuli from the ears, eyes, skin, and muscle. So if you have lights constantly flashing or noise constantly going on, it makes it hard to fall asleep. And its headquarters is in the midbrain. Motor function is involved here with maintaining muscle tone. The cerebellum, the second largest part of the brain, it does have a large amount of neurons for its size, so it's 1/10 of the mass, but half the neurons. This is going to fine-tune conscious and subconscious movements and helps adjust postural muscles. So ataxia would be difficulty with balance. This would result from damage from trauma or a stroke. Intoxication from alcohol or other substances can cause temporary disturbances in this. So our structures, the fovea is the surface of the cerebellum. It's a highly folded neural cortex. So you can see very squiggly-like appearance there from all the folds. The vermis is the central band of the cortex. So vermis actually means worm. Then you've got the anterior and posterior lobes that are separated by a primary fissure. The cerebellar hemispheres are separated at the midline by the vermis. The flacconodular lobe sits below the fourth ventricle, and the arbor vitae is the highly branched internal white matter of the cerebellum. So the arbor vitae you can see in here. So arbor means tree. So the Purkinje cells, they're large branched cells in the cerebellar cortex. They can receive input from up to 200,000 synapses. The cerebellar nuclei are embedded in the arbor vitae, and they're going to relay their information to the Purkinje cells. Our diencephalon is going to integrate sensory information and motor commands. This is going to include the thalamus, the epithalamus, which is also called the pineal gland. This will secrete melatonin, and the hypothalamus. Now, here's some more pictures of the brain. And then we've got a cross section here for the frontal section. So here you can see where the third ventricle is surrounded by the diencephalon. So the superior part of the walls is going to be your thalamus. The inferior part is the hypothalamus. So the thalamus is a big relay station. It's going to take sensory impulses, except smell, and motor impulses from the cerebellum and basal ganglia to the primary motor area of the cerebral cortex. It does help play a role in consciousness. The anterior nucleus plays a role in emotion and memory. The medial nuclei play a role in emotion, learning, memory, and cognition. The lateral group functions in emotions and integrating sensory information. The ventral group is going to have various relays, plays a role in movement control, somatic sensations, visual and auditory impulses. The intralaminar nuclei play a role in arousal, and they'll integrate sensory and motor information. The midline nucleus is presumed to function in memory and olfaction. And then the reticular nucleus filters and integrates impulses from other thalamic nuclei. So this just lets you see where you've actually got the thalamus inside the brain. So these are deeper in. And this is all of those different nuclei that you have in there. So your hypothalamus is inferior to the thalamus. It's going to consist of a mammillary body, a median eminence, infundibulum, and nuclei. It will control somatic motor activities at the subconscious level. So it will control autonomic function and helps to regulate body temperature. It's going to coordinate activities of the endocrine system and nervous system. It's going to coordinate circadian cycles of activity, so your day and night cycles. It's going to produce emotions and behavioral drives. So you've got a feeding center in here and a thirst center, things like rage, sex drive, fear, pain, pleasure. It coordinates your voluntary and autonomic functions, and then it will secrete hormones. So a lot of these are going to be the releasing hormones that will trigger the pituitary to release other hormones, as well as ADH and oxytocin. All right, so the pituitary is important because it controls a lot of other endocrine glands. It actually sits in the sella turcica, which is a little well in the skull, and the optic chiasm sits on top of it, so or crosses it. So you can have where you would have a pituitary tumor, it can actually start to affect vision with the optic chiasm. So Epithalamus, or your pineal gland, it's an endocrine gland about the size of a small pea that secretes melatonin during darkness. This helps to promote sleepiness and help set your biological clock. And then the Haber nuclei, this is going to help with your emotional responses. This is where you have emotion and olfaction tied together. So the subthalamus and circumventricular organs. The subthalamus is an area just inferior to the thalamus, and it's going to work with the basal ganglia, cerebrum, and cerebellum to help control body movements. Your circumventricular organs are in the walls of the third and fourth ventricles. They are going to monitor changes in the blood chemistry because that area lacks a blood-brain barrier. It's part of the hypothalamus, pineal, and pituitary gland. This is how HIV virus can actually enter into the brain, leading to dementia. Your limbic system, this is the emotional aspect of the brain. It's a ring of structures on the inner border of the cerebrum and the floor of the diencephalon. So this functional grouping helps to establish your emotional state. It will link conscious functions of the cerebral cortex with autonomic functions of the brainstem, and it will help facilitate memory storage and retrieval. So some components that are parts of the limbic system. The amygdala deals with anger, danger, and fear responses. It interfaces the limbic system with the cerebrum and sensory systems. The limbic lobe of the cerebral hemisphere is going to have the cingulate gyrus. It plays a role in expressing emotions through gestures and helping to resolve mental conflict. The hippocampus, it will convert short-term memory to long-term memory. And then the memories get charged with emotion are often retained more. The fornix is a tract of white matter that's going to connect the hippocampus with the hypothalamus. The anterior nucleus of the thalamus is going to relay information from the mammillary body to the cingulate gyrus. And then the reticular formation is going to be involved with stimulation or inhibition effects of emotion. So things like rage, fear, pain, sexual arousal, pleasure are all involved in here. So you can see where all of these structures are. This is a sagittal section. So our cerebrum is the largest part of the brain. It's considered the seat of intelligence. So your cerebral cortex, the surface layer is 2 to 4 millimeters thick, and it's made up of gray matter with billions of neurons. So we have the gyri, which are the raised areas or the convolutions, and then the deep grooves are the fissures, and the shallower are the sulci. So you have cerebral white matter that's beneath the cortex. This will have tracts that are connected to parts of the brain within itself and other parts of the nervous system. So it's nearly separated into right and left halves that we call hemispheres by the longitudinal fissure. It is actually connected on the inside by a bundle of transverse white fibers called the corpus callosum. So the three main functions of the cerebrum, each cerebral hemisphere is going to have sensory information from the opposite side of the body, and it's going to send motor commands to the opposite side of the body. So the two hemispheres do have some different functions. Their structures look alike, but their function is slightly different. So it's important to have the correspondence between these two sides of the brain to allow everything to function. The correspondence between a specific function and a specific region of the cerebral cortex is not precise. So we can't look and say, okay, this part of the cerebral cortex is damaged. This is exactly what function is damaged. It's not always really straightforward mapped out. We have an idea of what's in that area, but the exact location, we are not able to see at this point. So with hemispheric lateralization, you have functional differences between those right hemispheres. In most people, they're going to have the left brain be a little bit more dominant hemisphere. It doesn't mean that you don't use the right brain, it's just the other side slightly more dominant. So that's how it controls reading, writing, math, decision-making, speech, and language. The right cerebral hemisphere is going to relate to sense, like touch, smell, sight, taste, feeling, recognition of faces, voice inflections. Sometimes when there's unclear dominance, it may actually contribute to dyslexia. So this just gives you a nice little comparison between the two hemispheres in there. Like I said, it's not that you use one and not the other. You just may have more of a preference for one over the other. So when we look at the lobes and fissures, you have the longitudinal fissure that should be in these colors are not coordinated the way it says on here. The longitudinal fissure, you've got, it's going to run longitudinal on here. You've got the frontal lobe here. The central sulcus is going to run across. So you've got the precentral and postcentral gyrus in front of it and behind it. The parieto-occipital sulcus back here. And then you've got your occipital lobe in the back, and the lateral sulcus in blue. And the temporal lobe is way down here. And the insula you cannot see on here. Oh, here we go with the lines. Now you can see where the insula is deeper in the brain. So when we look at the cerebral white matter, we have these association tracts or fibers that are going to conduct the nerve impulses between the gyri in the same hemisphere. The projection tracts or fibers are going to conduct nerve impulses from the cerebrum to the lower CNS. And then the commissural tracts or fibers are going to cross the hemispheres. So this would be things like the corpus callosum and the anterior and posterior commissure. So here you can actually see these fibers in here, projection fibers from the descending and ascending tracts, commissural fibers from one hemisphere to another, and association fibers between the gyri in the same hemisphere. And then we can see more of the fibers here in red. So projection fibers pass through the diencephalon, the brainstem, the cerebellum, and spinal cord. And then we have your ascending and descending projection fibers. So within each hemisphere, you have arcuate fibers. They're short fibers that will connect one gyrus to another. The longitudinal fasciculi are longer bundles. They're going to connect the frontal lobe to the other lobes in the same hemisphere. And then your commissural fibers connecting the two hemispheres, corpus callosum, anterior and posterior commissure. So the basal nuclei, you've got masses of gray matter embedded in the white matter. They will direct subconscious activity, so they're involved with the initiation and termination of movements, subconscious control of the skeletal muscle tone. So when you're sitting there, you're not flaccid. It helps to maintain some muscle tone of things. And then the coordination of learned movement patterns like walking and lifting. And this just allows you to see some of these images. So when we look at the sensory areas of the cerebral cortex, you've got the primary somatosensory area. It's in the postcentral gyrus. This is going to receive information on touch, pressure, vibration, itch, tickle, pain, temperature, proprioception. Your primary visual area is in the occipital lobe. Primary auditory area is in the temporal lobe. Primary gustatory area is in the insula and parietal lobe. And the primary olfactory area is in the temporal lobe. So here you can see that you have association areas with things. You can have Wernicke's area that's involved with speech, Broca's areas that's involved with speech. So we have the primary motor area. This is going to be in the precentral gyrus. That's going to control motor functions that are under voluntary control. Broca's area is involved with speech production. So functional organization in the association areas. Your somatosensory association area is posterior to the primary somatosensory area. This allows us to experience the shape and texture of an object by feel and memories associated with the feelings. The visual association in the occipital lobe, the auditory association in the temporal lobe. We have facial recognition, auditory association, orbitofrontal cortex like olfaction in that area. In Wernicke's area, it is predominantly in the left temporal and parietal lobes. This has to do with speech and language interpretation. The prefrontal cortex, the anterior portion of the frontal lobe, here is where you're going to have personality, intellect, complex learning, the recall of information, initiative, judgment, foresight, reasoning, intuition, mood, several of those types of things. In the common integrative area, this is in the somatosensory area, visual and auditory areas. Your premotor area, this is where you have your learned motor activities of complex and sequential nature. And then the frontal eye area controls the voluntary movement to the eyeball. So this gives kind of a nice, simple summary of what goes on in these cortical areas. So when we look at the electroencephalogram or EEG, this is measuring the brain activity. So a flat EEG is evidence of death clinically. Alpha waves, these are normal to see in a healthy resting adult. Theta waves are normal in children. Beta waves are what you would see in a concentrating adult. So hopefully those are what's going on during a test. Delta waves are seen during normal sleep. So decussation, this is the crossing of the nerve fibers. So we have the pyramidal decussation that marks the border between the spinal cord and the medulla oblongata. What's significant here is when you have, because of this decussation, clinical presentation will be on the opposite side of the body from where the actual damage is. The lateral corticospinal tracts contain over 90% of the fibers present in the corticospinal tract, and they run the length of the spinal cord. The primary responsibility of the lateral corticospinal tract is control the voluntary movement of the contralateral limb. So ipsilateral is the same side, contralateral is the opposite side. So when we look at brain injuries, concussion is a mild traumatic brain injury. This can occur after impact to your head or a whiplash type injury. So sometimes they're called an acceleration deceleration injury. It causes your head and brain to shake. Basically, the brain slams against the head on the inside. It can result in an altered mental state that may include becoming unconscious. TBIs is the abbreviation for a traumatic brain injury. It's injury to the skull. These may be permanent, and the consequences are a lot more unpredictable. Since it varies with location, mechanism, and severity, every injury tends to be a little unique. Cerebrovascular accidents or strokes. Here, blood flow to part of the brain is either stopped by a blockage or a rupture of a blood vessel. So without blood store being returned, those areas are going to die eventually. So transient ischemic attacks, they're often called a mini stroke. It's a temporary blockage of flow to the brain. It does not cause permanent damage, so a lot of times people ignore it. This can be a real important warning sign that a full-blown stroke is coming. So ataxia, it's impaired balance or coordination. This can be due to damage to the brain, nerves, or muscles. There's a lot of things that can cause ataxia. Aphasia is an acquired communication disorder that impairs the person's ability to process language. It's not an intelligence issue. They just are not able to convey what they want to in speech or understand other people's speech. And this can result from strokes frequently.